faculty member. I originally worked in the industry, so I teach evenings at City College, and I've been teaching there since '97. And I've been involved in about 3 startup companies, and after that I went and did a PhD and came back last year. And when I came back last year, I wanted to do something different besides working at a startup company. So one of the first things is teaching in this stem cell technology program. So the person that really should be presenting this is Karen or Edie, but they've been busy, so I'm doing it on their behalf. So the objective of the stem cell certificate program is to train individuals with this specialized skills that are needed for the growing field of stem cell research. And this was a program that was launched in 2006. And the picture that you see there is actually an actual stem cell CCF student. So this is the course structure. The prerequisite is 1 semester biology with lab and 2 semesters of chemistry, including organic with lab. The certificate program revolves around 3 main courses: Biotech 21A, which is the mammalian cell culture; Biotech 21B, which is fluorescent cell technology; and 21C, stem cell technology course. And all 3 are done in chronological order, so that by the time they get to the stem cell technology course, they're well versed with cell culturing. So you can see the time frame there. Mammalian cell culture is about 6 weeks, 9 hours per week. The fluorescent cell technology is 6 weeks, 9 hours per week. And the stem cell technology is 8 weeks, 9 hours per week. Students also elect to go on and take more advanced courses, including PCR, ELISA, and Western blotting. We really do not have a text, but we generally refer to Ian Freshney's culture of animal cells. The classes are offered in fall, spring, and summer, day and night classes. So what this does is that it gives the students about a year to complete the program. So looking at the course, the main idea of mammalian cell culture is basically to get the students working with cells. They get to know all the equipment that is necessary to culture cells, the CO2 incubators, laminar flow hoods, how to detect contamination, how to plate cells, how to count cells, how to do growth curves, and so on. In fluorescent cell technology course, this is where they actually label cells, but even more important is that they get to know the principles and the operation of the epifluorescent microscope. And by the time they get to stem cell technology, the main technology that they get their hands on is the fluorescent activated cell sorter. How does this go? How do you move this to the next slide? Yeah, that's true. And it doesn't work either. There we go. So here is our stem cell culture facility. This is where we teach both the mammalian cell culture as well as stem cell technology and fluorescent cell technology. This is a room that's about the size of this room, and you can see it has all the laboratory equipment spread out, the laminar flow hoods, and in the middle we have the culture counters, and what you don't see towards the left is the fluorescent activated cell sorters. On the top you see where the lectures are held. So this is an ideal way of teaching the courses, because you can lecture, and if you want to demonstrate something in the laminar flow hood, you can take all the students there and demonstrate that. Okay. Now, the instrumentation, here is our 2 flow cytometers. There are BD fax scans. We also have 6 epifluorescent inverted microscopes, 10 inverted microscopes, 6 CO2 incubators, and 6 laminar flow hoods. I mention these instruments because our general class size is about 20 students, and we train them pretty much on an individual basis using these instruments, because these instruments are crucial to do stem cell research. Okay, one thing I don't have is that I wanted to put the curriculum for all 3 courses, but I kind of felt that was not really necessary, but this is what we want the student to get out after he gets the stem cell technology certificate. Basically, to understand the principles of stem cell biology, we give them a good introduction to the theoretical principles as well as practical principles, to critically and independently assess the reality and future of applied stem cell biology. And we do this by actually giving them current literature for homework assignments. And what they have to do is they have to read this literature, and they'll be questioned either on an exam or on a surprise quiz. To differentiate between different types of stem cells in terms of their function, use, and care. Isolate mesenchymal and hematopoietic stem cells from mouse bone marrow. And we don't just give them a bunch of bone marrow and give them the technology to isolate, but we actually give them the animal, the mouse, where they have to dissect out the femur, in a good amount of time, and then isolate the bone marrow, then proceed to culture them and use a technology like MAX, which is magnetic activated cell sorting, to isolate the hematopoietic stem cells or mesenchymal stem cells. Of course, we also we give them embryonic stem cells, and the embryonic stem cells have worked with our mouse system because we're not certified for the human embryonic stem cells. So they're taught how to form embryonic bodies and how to differentiate them into target tissues such as cardiomyocytes and skeletal muscles. And the last 2 is basically to understand and operate the flow cytometer and analyze flow cytometric data. The last 2 is basically a course in itself because it takes a lot of time to understand. Okay, after they go through the courses, then some are chosen for an internship program. And actually they're paid for this. I think they get about $2,000 a month. And they're sent into various research labs in the San Francisco Bay Area, and some are UCSF and Genentech. Today we have about 150 students that have received the certificate, and approximately half have been hired at UCSF, Stanford, Gladstone, even prestigious stem cell research places like Geron. So the stem cell program just didn't appear, but it slowly evolved over time. And here's a timeline that shows that from 2000 to 2009. And what really helped was the Proposition 71 initiative, the creation of CIRM, which is a California initiative for regenerative medicine, that gave us some money to buy equipment, and also gave us the CIRM grant in 2009. So we have the stem cell program now, but it doesn't stop there. So what we're doing right now is that we're working with a principal investigator at Stanford University, Christopher Scott, and developing a curriculum that could be nationally implemented. The aims of this curriculum is basically to build a multidisciplinary undergraduate program for community and four-year colleges. It's cross-disciplinary, that is, both the applied science, lab skills, ethics, law, and policy are incorporated. And the first pilot module goes into effect in about a month, actually, in my class. And it also has quality measurements on how well the student is absorbing the material. So the course is being developed for over a year now, and here are the content developers, and they're basically spread out across the country. So 1 year in progress, so we have about 80 hours of instruction and materials, and they're basically like primers, which has a short topical introduction for the student. It also has slide presentations and fully illustrated sets with teacher text and image library. It also has activities for the students in order to measure how well they're learning the material. And it also has fully immersive lab teaching skills.